In this video we will discuss about how this inhibition of BH2 or the pro-survival proteins cause activation of p53. So we will not have the answer about how this is occurring, but we will find evidence that inhibition of BH2 or pro-survival proteins can cause activation of p53. So let us understand how they come up to this finding. So in order to test this finding, they used two different kind of cells, this AF47A and 214 DHL, and in order to inhibit this BH2 BH2 family proteins, in the case of AF47A they use these inhibitors S63845, and they use also a different cell lines, that is 560, but they did not mention too much about the results of this 560. So we will not cover these results, as these results are similar to what we will cover for this AF47A and 214 DHL. So for this 214 DHL cell lines, they use this venetoclax to inhibit the BH2 survival proteins. And what they have find out is that when they inhibit these BH2 survival proteins using this the inhibitors, they can see the activation of p53 after 4 hours in AF47A, and in this 214 DHL cell lines they see the activations usually after 6 hours. After 6 hours the signals seems quite clear. And in order for control, what they did is they directly activate these p53 cell lines, p53 proteins using this nuclein 3A. And in all the three cases, what they have find out is that p53 activation occurs after 1 hour. This indicates that there could be some indirect relationship between these BH2 survival proteins with this p53 activations. That's why the p53 activation occurs late. And in order to further validate their points, what they did is they use these p53 knockout cell lines and the wild type cell lines, and then they did the RT-PCR or quantitative qPCR, quantitative PCR or qPCR, and check the genes that are upregulated by p53 after treatment with this S63845. And what they find out is that when they use the normal or where p53 is not mutated, they see an increase in the markers such as NOXA, PUMA, P21, BAME. When they use knockout cell lines, they see a relatively low increase in those markers. So, and more specifically for this PUMA and P21, we see a significant decrease in those increase in the markers. For this AF47A, it is quite evident for PUMA and P21, and for 560 it is quite evident for this P21. So after confirming, so this results further confirm that the p53 activations occurs with the inhibition of this BH2 survival proteins using S63845, and they did analysis at different time intervals that is shown here. And thereafter what they did is they also did the gene set enrichment analysis of these samples after the 24 hours of this S63845 treatment, and they have find out is that compared to the p53 knockout conditions, the wild type cells treated with these inhibitors, BH2 pro survival proteins inhibitors, shown an increase in the p53 pathways. And when they check the individual genes increase, that individual genes that are increased individual genes that are the markers of this p53 upregulations. They have find out that in the case of control, they see an increase in those markers such as PMAP, PMAP1, DVC3, CDKNA1, BCL2, L11. They see those increase in the markers. But where the p53 is knocked down, the increase in the markers is very low or almost negligible. This analysis they did it on this AF47A cells, and they did the similar analysis on this DHL cell, 214 DHL cells. Here they presented results in Western blot, where we can see that the when the p53 activations is occurs when this inhibitors of BH2 survival proteins is there in both the DHL 214 and DHL 216, and the markers are also shows the similar result that the beam will activate when these BH2 survival proteins are inhibited in both the cases, and these things are quantified on the panel F, where we can see that p53 increase in the p53 PUMA BIM BCL2 occurs with the inhibition of BH2 families. BCL2 is not occurs, but for the other things, the markers, we can see that increase, but they did not show the result for the whether these are increases statistically significant or not. This could be the criticism about this paper. Thereafter, they also did the analysis on this knockout cell lines of this DHL, and here they inhibit. So this is I think this is also for this AF47A cell lines, not for the DHL cell lines. So they did the analysis on AF47A cell lines, and what they did is uh, they use these inhibitors S63845A in the control, and they see a slightly increase in the p53 cell lines, but for the TP53 they do not see any increase in the cell lines. And for this DOHH2, they see the similar results, that is in the control case they see an increase in the TP53, but when the TP53 is knocked down, they do not see any increase in the signal for the TP53. So these results are not on these cell lines, neither in AF47A or 214 DHL. They use a different cell line, that is BL2 or DOHH2, which is quite similar to the leukemia or blood cancer cell lines, which are derived from them. And for this DOHH2, they see the markers impact, and they see that in the case of control, they increase in the markers for the p53 genes such as NOXA, BBC3, PUMA, and P21, but do not see a significant increase in this TP53, or there is a less increase in the markers for this P53 TP53 in the case where T53 is knocked out cell lines is used, and that results are shown here for that, such as this NOXA, PUMA, P21. So all this indicates that the inhibition of BH2 proteins somehow indirectly in the panel I, it is shown that when they inhibit these pro-survival proteins, the pro-apoptotic proteins goes down, and what will happen is that there is a activations of the p53 occurs, and that will further accelerate this process. So how this BH3 mimetics cause this activation of p53, they don't know, but from the results they know that there is a indirect relationship between p53 mimetics and p53 activations. So that's all for this video. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.